With the identification of Z3 and Z4 in 1986 and of Z9 in 1992, the worm C. elegans had been established as a model for apoptosis studies. David Rowe, who had demonstrated in 1988 that human BCL2 can block apoptosis, became interested in determining whether the expression of human BCL2 in C. elegans similarly could block apoptosis. Why did he want to do that? While C. elegans genes had been identified whose functions are required either for blocking apoptosis or for inducing apoptosis, the genes had not been cloned yet and their gene products were therefore unknown. For this reason, it was unclear at the time whether the apoptotic process that investigators like David Rowe were studying in mammalian cells and that investigators like Bob Horwitz were studying in C. elegans were really the same processes and conserved at the molecular level. The question basically was whether similar proteins and molecules were involved in the apoptotic process in mammals and worms. To test whether the expression of human BCL2 in C. elegans could block the death of the 131 cells that normally die during C. elegans development, David Rowe and colleagues at Stanford University generated a C. elegans line transgenic for a reporter construct that allowed the expression of the BCL2 transcription unit under the control of a heat-inducible promoter. Using a shift in temperature, he could then induce broad expression of BCL2 in C. elegans. How do you generate transgenic lines in C. elegans? The traditional way is the following. You take your plasmid of interest and a co-injection marker, a plasmid that expresses something that you can easily detect or assay for. And you inject these plasmids into the gonads of hermaphrodites. Inside the gonads, the plasmids will be taken up by some of the oocytes that are generated. In these oocytes, the plasmids are made into what is referred to as extrachromosomal arrays. Extrachromosomal arrays are like mini chromosomes that can contain anywhere from 30 to 300 copies of the injected plasmids. In general, one therefore looks at the overexpression of the plasmid and therefore transcription unit of interest. Since the extrachromosomal arrays do not have what normal chromosomes have, such as centromeres, they do not always get segregated properly into the daughter cells during cell division. They often get lost. That's why only a few of the progeny generated from the injected animals will actually carry the extrachromosomal arrays, and not all cells in these animals will actually carry it. The animals are therefore mosaic. Furthermore, only a few transgenic F1 animals will carry the extrachromosomal array in their germline and will therefore pass on the extrachromosomal array to some of their progeny. Those clones that do are referred to as transgenic line. I should mention that nowadays there are other ways to make transgenic C. elegans. Low copy arrays, for example, can be generated by bombarding C. elegans with a gene gun, and targeted single copy integration into the genome can be done using a transposon-based method. Now back to the expression of human BCL2 in C. elegans. David Rowe actually used Z1 mutant animals to generate the transgenic lines. Why did he use Z1 mutant animals and not just wild-type animals? He used Z1 mutant animals for the same reason that Hilary Ellis and Bob Horwitz had used Z1 mutant animals for their first screen. In an engulfment defective background, apoptotic corpses persist for a long time, and you can therefore analyze and quantify cell death that occurred over an entire developmental period, for example through embryogenesis, and not just at one particular time point. And what David Rowe observed in transgenic animals, which he had shifted to a higher temperature to induce BCL2 expression, is a significant reduction in the number of persistent apoptotic corpses. In animals that carried a control plasmid, or in animals that were not shifted to the higher temperature, he found on average 40 apoptotic corpses. However, in the animals that carried the BCL2 plasmid and that were shifted to the higher temperature, he found on average only 16 apoptotic corpses, less than half the number. What could explain the lower number of apoptotic corpses? It could be the result of fewer cells dying. It could reflect a decrease in apoptosis. However, it could also reflect the suppression of the SED1 engulfment defect. To rule out this possibility, David Vo analyzed how long the corpses that he detected in the BCL2 expressing animals persisted. He found that they persisted as long as in SED1 control animals. This suggested that the lower number of corpses seen in the BCL2 expressing animals was a result of a decrease in the number of cells that had died. Basically, many of the 131 cells that normally die during C. elegans development no longer died. This finding, which was published by Vo and colleagues in the journal Science in 1992, shortly after the identification of SED9 was published in the journal Nature, was very significant. It was significant because it demonstrated that the human BCL2 protein can block apoptosis in C. elegans. Since proteins don't act alone, but in networks and pathways, it furthermore suggested that a pathway exists in C. elegans in which the BCL2 protein can act to block apoptosis. This finding was therefore the first hint that an apoptosis pathway exists that is conserved from worms to humans. BCL2 can block apoptosis in mammals. Similarly, SED9 can block apoptosis in C. elegans. BCL2 can also block apoptosis in C. elegans. In their discussion, Vo and colleagues state the genetic behavior of SED9 is the most similar to that of BCL2. They do not go as far as proposing that SED9 might be the ortholog of BCL2. However, they do speculate that as SED9 appears to act by inhibiting SED3 and SED4 activity, BCL2 most likely works in humans by antagonizing the activity of homologs. Do the BCL2 and SED9 genes encode similar proteins? The SED9 gene needed to be cloned. And this was done by Michael Hengartner in Bob Horwitz's lab, who had identified and characterized the SED9 mutants.